Hi students, this is Dr. K. Shivaya, Department of Pharmacology, Faculty of Santiram College of Pharmacy. Our next topic is formation of ATP. Already you know very well regarding how the ATP is formed. Already earlier classes, nothing but in inter or 10th or in 10th, you studied how the formation of ATP. So the glucose mainly to form the ATP, but how it is? So the glucose undergoes aerobic respiration and anaerobic respiration to produce the ATP. See, if you take in the aerobic respiration, the 6 oxygen molecules with glucose molecule after reaction takes place to produce the 6 carbon dioxides, 6 water molecules and ATP. So first we have to know about the what is adenosine triphosphate. See, this is adenine molecule, this is ribose molecule, and in that have the 3 triphosphates, 1, 2, 3. If one phosphate, if it is break to produce the energy, so ATP is the energy cell can use to do work. Like currents, how the currents you are using for to buy, but here if the cell want to work, it's compulsory needed the energy, that is in the form of ATP or phosphate. See, energy is released when the phosphate bonds are broken. See, know this slide already, you know. See, these ADP plus phosphate, it is formed. This is a substrate formation. We can see next slides. So the ATP is formed. So he is absorbed energy, but when ATP utilized, the one phosphate is release energy, that is adenosine diphosphate. You know that the glucose converted into ATP in the 3 steps: glycolysis, Krebs cycle, electron transport chain. See, the glycolysis takes place in the cytosol, Krebs cycle in mitochondria, electron transport chain in inner layer of the mitochondria. Okay. So what is, see, this is important, we have to think and take notes. What is ATP? Where is the energy stored in ATP? And what are the 3 steps needed to turn glucose into ATP? You know that. So what is ATP? ATP is the only molecule that the cell can use for energy to do work. Where is energy stored in ATP? Energy from ATP is stored in the phosphate bonds. So one phosphate bonded with another phosphate, like that the 3 phosphate bonds. So like there it is stored. Where the 3 steps, where it is formed: glycolysis, Krebs cycle, electron transport chain. So glycolysis, it is takes place in the cytosol. So here no oxygen required. So when glucose means after glycolysis, end of the glycolysis, we can get 2 pyruvate net of 2 ATP. Actually 4 ATPs come, 2 ATPs utilized. So the net 2 ATP, and along with 2 high energy molecules are produced, that is 2 NADH are produced. So this is entire regarding about glycolysis. So it is occurs in cytoplasm. It is not required for not required oxygen. So after glycolysis, one glucose is converted into 2 pyruvate plus 2 ATP plus 2 high energy molecules. Next cycle. So at the end of the glycolysis, the pyruvate is formed. You know, that what is the Krebs cycle. This is one of the scientists. This also we can call a citric acid cycle because of the first starting substrate in the cycle is citrate. That's why it is called as a citric acid cycle. So here the pyruvate is converted into acetyl coenzyme A in the enzyme of pyruvate dehydrogenase. Here the NAD plus is converted into NADH and H plus. NAD plus and NADH and H plus. Next, these acetyl coenzyme A to form the citrate, in the presence of citrate synthetase. So here the removal of coenzyme, then again the coenzyme reutilized. Then these citrate in the presence of aconitase to form the isocitrate. Then isocitrate to form the alpha ketoglutarate in the presence of isocitrate dehydrogenase. So dehydrogenase meaning of removal of hydrogen molecule. So that's why here the NAD plus is converted into NADH and H plus. Here also we can observe the pyruvate dehydrogenase removal of. So here the removal, and here adding of NADH and H plus. So that is dehydrogenase enzyme. Then after the alpha ketoglutarate again to form the succinyl coenzyme A. Again here that also alpha ketoglutarate dehydrogenase, nothing but whatever the contain the hydrogen molecule in this pyruvate removed and isocitrate in the contain hydrogen removed, again here it contains the hydrogen here removed. So that's what to form the succinyl coenzyme A. See the succinyl coenzyme A after formation from the alpha ketoglutarate to form the succinate. So here the succinate coenzyme A synthetase, simply we can call succinate synthetase also. Next, the succinate is converted into fumarate in the presence of succinate dehydrogenase. Here also removal of hydrogen from succinate to form the fumarate. Then fumarate in the presence of fumarase enzyme to form the maltase. Here addition of water. Then maltase is converted into oxaloacetate. Malate is converted into oxaloacetate in the presence of malate dehydrogenase. Here also the removal of a hydrogen molecule. So that's why NAD plus and NADH and H plus. So if you observe where the removal of one hydrogen molecule from the substrate into the state, it is formed the NAD plus 2 NADH and H plus. Here also NAD plus NAD plus. Here also. So how many here in the citric acid cycle? 1, 2, 3. So between the pyruvate and citric acid cycle 1. So total here maintain 4. Also here also one FADH2 is also formed. This is the citric acid cycle. So the Krebs cycle, it is occurs in the mitochondria. So here it is requires oxygen. So in this the 2 pyruvates. So in the after glycolysis, so you can get 2 pyruvates. That's a 2 ATPs. 8 high energy molecules NADH and FADH2 are formed. So electron transport chain. See the electron transport chain, it is occurs in the mitochondrial cell membrane, that is the inner membrane. So it is requires oxygen. So 10 high energy molecules to transport the protons. So after these 10 high energy molecules to transport the protons and the electrons to form the 30 ATPs. So now ATP count is 34. So first it is glycolysis 2 in Krebs cycle, again 2, here again 30 ATPs. So net count is 34 ATPs. So glycolysis Krebs cycle, so the one glucose molecule to produce is 34 ATPs. Actually 36 ATPs, so 2 ATPs are utilized in glycolysis. So that is the come to the anaerobic respiration. So here there is no oxygen, anaerobic takes place. So only glycolysis can take place, which makes 2 ATP. So what we can observe? Two kinds of anaerobic respiration. In animals produces 2 ATP plus lactic acid. So in yeast produces 2 ATP plus ethyl alcohol. That's only 2 ATPs. So in animals, in human beings also lactic acid is produced. So these lactic acid to produce the muscle cramps. That's why if you run more, then automatically the oxygen levels decreased in the muscle, then automatically the anaerobic respiration takes place in the cell. That's why lactic acid released. That's why you can get the pain in the legs. So if you run more, that's the thing. So aerobic respiration have oxygen. So what do you do on a daily basis? Like as these are the makes 34 ATPs. So these are the overall anaerobic respiration, aerobic respiration. Here no oxygen, here oxygen. Here makes lactic acid and alcohol. Here the 2 ATPs only produced. Here the 34 ATPs are produced. Net, I am telling you, net 30. Here net. Here overall actual 36, net 34 ATPs. Then if you take about the formation of ATP substrate level, involves the phosphorylation of ADP to form ATP. So you know the adenosine diphosphate is a secondary messenger. It is present in cytosol. So when you take up the another phosphate to form the adenosine triphosphate, adenosine diphosphate, it also contains adenine ribose plus 2 phosphate bonds. When it is bound with another phosphate to form the adenosine triphosphate. So where it is found, we can check it. So for few examples, see the conversion of 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate. So here the phosphate molecule removed. So in the presence of phosphoglycerate kinase, the maximum involved in the phosphorylation in glycolysis also. So that's why ATP. Again the co-molecules, the magnesium 2 plus ions are involved in the formation of ATP. So the ADP combined with magnesium 2 plus ions with the help of the phosphoglycerate kinase to form the ATP. So this is the formation of one of example. Another example. So this is substrate level. So conversion of phosphoenol pyruvate to pyruvate. So it contains one phosphate. So this is phosphoenol pyruvate is converted to pyruvate in the enzyme pyruvate. Here also magnesium 2 plus. I had said that the ATP utilize the magnesium 2 plus ions during the formation of ATP. The adenosine diphosphate utilize the magnesium 2 plus ions in the formation of ATP. It's the key. These are the few examples. Succinyl coenzyme to form the succinate. Here also ATP formed. So this is comes under the Krebs cycle. Krebs cycle and creatine phosphate. So this is in the presence of creatine kinase. We can discuss in the metabolism of creatine. So it is a equal distribution. If the creatine phosphate is more, it is converted to creatine. If the creatine and more, if it is converted to creatine phosphate. So it is equilibrium. So that's why it is a 2 given. So equilibrium. So this is regarding about ATP formation. Next we can discuss about basal metabolic creatine metabolism. This is regarding the creatine metabolism. So what is the creatine and creatinine? So in your syllabus they given the creatinine. Creatinine coming from the creatine. See, actually the creatine and creatine are not the same substance. Creatine is found in the muscles. Creatine is a breakdown product of creatine phosphate. So these coming from the creatine or creatine phosphate. So the creatine, if taken the creatine is an amino acid, it is found in proteins. Creatine is a nitrogenous organic acid. So how the creatine is synthesized? It is required 3 amino acid: glycine, arginine, methionine. Glycine, arginine, methionine. This is a next. These are the major sites for the synthesis of creatine is kidneys and liver. See how sites of creatine synthesis. So if you take an ornithine and arginine, see creatine synthesis requires 3 amino acids: methionine, glycine, arginine, and 2 enzymes, that is arginine glycine aminotransferase. This is arginine glycine aminotransferase. So because this enzyme to form the guanidinoacetate acid, guanidinoacetate acid. So this is a GA. So it is enter into the blood. Then again the here it is enter into the liver. In the liver, in the presence of guanidinoacetate methyl transferase. So this is here. This is not written well. So here we have to take guanidino. So whatever the formation of a guanidinoacetate acid, it is undergoes methylation. It undergoes methyl transferase, in the presence of methyl transferase. So which methylates to produce the creatine. Produces the creatine. These creatine again enter into the blood and excreted through the another form of in the kidney. So this is the how the formation of kidney. So through the again, if you want to listen again, you go back to my video. Next, this is a metabolic burdens of creatine synthesis. So how so 70 millimoles of glycine required, 37 millimoles of arginine required, and 90 millimoles of methionine required. 70. Adult male ingest 80 grams of protein per day. Synthesis of 8 millimole creatinine per day could potentially use. So in that, so these are the calculation part. So distribution of the creatinine in the body. So from the liver, the transported to the other tissues. So because of I already said that the creatine it is formed. It is started from the kidney. Then after that it is entered into the liver. Then after formation of creatine again enter into the blood. Then from the blood it is entered into the kidney. So the 98% of creatine present skeletal muscle and heart muscle. This is very important. 98% of the creatine present in skeletal muscle and heart muscle. Okay. In muscle gets converted into the high energy source creatine phosphate. See here, the creatine is converted into creatine phosphate. So it's required one phosphate utilized. So already said that the formation of it is equilibrium. So creatine phosphate to creatinine we are discussed in the substrate formation of ATP. See, if the creatine is more, it is converted to creatine phosphate. If the creatine phosphate is more, it is converted to creatine. So this is equilibrium in the presence of creatine kinase. Creatine and creatine phosphate exists in the reversible. That's why reversible equilibrium in the skeletal muscle. In skeletal, approximately 1/4 of the creatine exists as a free creatine and 3/4 exists as a creatine phosphate. This is the form. So creatine phosphate is a high energy phosphate compound, acts as a storage form of energy in the muscle. So is a storage form of energy in the muscle. Already is discussed. So when the creatine is converted into creatine phosphate, one phosphate breakdown, phosphate it will break, energy produced. So provides a small but ready source for energy during the first few minutes of the intense muscular contraction. It's produced very small. Because of one ATP is converted into ADP plus one phosphate will come outside, that phosphate only it is utilized. So very few, one phosphate, that's why very few energy, very small energy, but it is enough for muscular contraction. See the formation of now creatinine. So creatine plus ATP in the presence of creatine kinase. Creatine kinase on the phosphocreatine plus ATP, a creatine phosphate. These phosphocreatine spontaneous. If it is spontaneous breakdown or removal of phosphate, it to form the creatinine. So the both are to form the creatine. So suppose if we take in creatine, spontaneous and non-enzymatic cyclization. So the formation of creatinine is a spontaneous process. So it is formed from the creatine or phosphate creatine. So if you take in phosphate, can remove the phosphate and produces the creatinine. If you take in from the creatine, it is a spontaneous non-enzymatic cyclization. This is the formation of creatinine. This is very important. The sequence is very important. So the creatine and the creatine phosphate spontaneously form creatinine as end product. Creatinine is excreted in the urine. So like that, already we discussed that the creatine formed. First it is formed the kidney, then after the end product is formed the liver, then again it's come to the blood, again it is entered into the kidney. So in the kidney it is formed and it is eliminated, is excreted in the urine. So these serum creatine is a sensitive indicator of kidney diseases. So serum creatine increases with the impairment of kidney function. If the kidney is impairment, then automatically the serum creatinine is increases. See how. So creatinine and creatine phosphate to form the creatinine. So it is these formed creatinine, it is entered into the blood, nothing but plasma, glomerular filtration. Because of the blood compulsory enter into the kidney, the blood flow enter into the kidney. So the blood contains the creatinine. The creatinine excreted through the glomerular, after glomerular filtration it is excreted through the urine. See here the one question. What is the fate of creatine that was produced from breakdown of creatine phosphate during the anaerobic science? So the creatine is a waste product of the phosphate and it will be excreted by the kidney in the urine at the rate of 1 to 2 grams per day. So creatine, these are the, you can understand if you read. So the next important point is the creatine kinase. See, the creatine CK is responsible for the generation of energy in the contraction muscle. Already we discussed the entire part this. But these creatine kinase in 3 forms, that's why it is called isoenzymes. Means they are spatial differentiated. Already what is isoenzymes. So CK creatine kinase, MM mainly present in skeletal muscle, CKMB mainly present in heart muscle, CKBB present mainly in brain. So why it is needed for? Because of serum that is creatine kinase is increased either skeletal muscle or heart muscle or brain. If the creatine kinase increased, the creatine kinase MM increased the skeletal muscle. The creatine and skeletal muscle affected. If the creatine kinase MB increased, heart muscle affected. If the creatine kinase BB increased, brain affected. So then the tissue damaged, either skeletal muscle tissue damaged, this enzyme increased, heart muscle damaged, this enzyme increased, if the brain is damaged, CKBB increased. So this is that is mainly the either affected injuries or damaged or myocardial infarction, this thing. So this is mainly for the diagnostic value. See, this is the entire what you discussed. What is the purpose of assaying for CK over the period of time? See, myocardial specific injury, 100% increase in MBCK within 4 hours. So that's what we discussed. So depending upon that diagnosis, they can identify the which muscle is affected. So normal serum creatine level 0.7 to 1.4 mg per deciliter, and the serum creatine level is 0.2 to 0.4 mg per deciliter. The amount of creatine excreted is proportional to the total creatine phosphate content of the body. Okay, so therefore it can be used to estimate the muscle mass. So serum creatine is a sensitive indicator of the kidney diseases, kidney function test. The amount of creatine in urine is used as an indicator for the proper collection of 24-hour serum sample. So the normal urine output of serum the creatine is 15 to 25 mg per kg. So this is a normal creatinine level in urine. So this is the regarding creatine metabolism. Thank you for watching. All of you follow my YouTube video and do the assignment as quickly as possible. Thank you.